Hi everyone, my name is Pratyaksh Rastogi and I welcome you guys to this session. And I am a NEET faculty at Academy NEET, and I deal with biology. So I teach both, that is botany and zoology. So for today, I have selected one discussion for you guys. But before that, I want to make sure that my video and my audio is clear to you. If there is any problem you are facing, then I am apologize for that in prior. So guys, I hope that you are on track with your studies. Okay, so I wish you best of luck. So for today, I've selected a topic, you know, I've selected a topic for you, which is microbes in human welfare. Okay, so in microbes in human welfare, we'll be talking about those microbes which are actually useful for the human beings. Okay, so guys, let's start. Okay, so if I talk about microbes in human welfare, so as I told you, we are only talking about those microbes which are beneficial, which are useful for human beings. For if you would have studied the chapter which is biotechnology, or any other chapter, so in that in that basically what we had read is that how microbes are harmful to humans or to plants or to anything else. But in this chapter, we are only going to discuss about those useful microorganisms which are actual, giving us benefits. Now you can just imagine, you would agree that microbes, there are millions, trillions of microbes which are available on earth, right? In soil, we can say, in water, in air, inside our bodies. Yes, they are also present in our bodies. For example, we have E. coli, that is Escherichia coli. It's a bacteria which is present in our intestine. So they are also present into our bodies. Also, whenever I'm talking, whenever I'm doing anything, there are hundreds, millions of microbes which are present around me. So I'm getting contact with with those microbes at every single point of time, right? So microbes are also present in animals as well as plants. Now next is, they are present even at sites where no other life form could possibly exist, sites such as deep inside the geysers. Now there are microorganisms which can actually live in the most extreme conditions, like most hot temperatures, like the hot springs, where the temperature is actually 100 degrees Celsius when the water starts to boil. Okay. Also, they are living deep into the soil. Okay. The deep into the soil where there is no oxygen, nothing, but they are living up there. They are situated into the layers of the snow. It means at the most extreme cold conditions. Also, they are living in highly acidic environments. So it means that the microorganisms, they can be found everywhere. Okay. So microbes, now let us discuss about those microbes which are actually useful in household products. For example, whatever we use in our daily lives into our houses, like we also have microbes which are beneficial in those terms. So what are those? A common example of those microbes will be the production of curd from milk. Even like you would have seen your mother who prepare curd from milk. So how they are able to do so? Because of the microorganisms such as lactobacillus and other commonly called lactic acid bacteria. Okay, so there are bacteria present like lactobacillus or other lactic acid bacteria which are responsible for preparation of curd from the milk. Now how are they able to do it? What they do is, now during their growth, the lactic acid bacteria produce acids that coagulate and partially digest the milk protein. So whenever we are preparing curd from the milk, so the proteins present into the milk, they are digested or they are degraded by the lactic acid bacteria, because lactic acid means they are producing that acid. Okay, now what is happening? A small amount of curd is added to the milk as inoculum or starter. Now guys, what is inoculum? Inoculum is something which we are using in order to accelerate the process, okay, or the starter. Because without inoculum, because we have to have some source in order to, for example, if we are preparing a curd from milk, then we have to have that bacteria present with us. And how are we able to do so? So because we have a previous curd, so we take some amount of the previous curd we prepared, and then we are putting that amount into the milk, which is functioning as inoculum or starter. Okay, That small amount consists of millions of lactic acid bacteria, which are suitable temperatures. Those bacteria multiply and they convert milk to the curd, which also improves the nutritional quality by increasing vitamin B12. So additionally, during the growth of the bacteria, the vitamin B12 production is also occurring. Okay. Now next to that, in our stomach too, now in our stomachs also, the lactic acid bacteria is present and it plays very beneficial role in checking disease causing microorganism, and that is why we always say that we have to have curd, we should have curd, because curd helps in detoxifying our body. What is the detoxification is? That is eliminating toxins from the body, and how we are able to do so? With the help of these lactic acid bacteria, because what their function is, whenever they go inside the body, they make the other microbes die which are harmful for the body. So now the second is the dough which is used for making food such as dosa and idli, also fermented by bacteria. Okay, so also whenever you also would have seen your parent that whenever we are preparing dosa or idli or anything, we have to prepare the batter for that even earlier, to the time. Why is it so? Because we need some fermentation which is done by the bacteria. It means during the fermentation the dough would have risen up, the dough would become puffy. Now how it is causing? Because of the production of carbon dioxide gas by that bacteria, and the process is called as fermentation. So during the process of fermentation, if you remember the chapter respiration in plants, and that I was talking about fermentation. So in fermentation, what happens? Whenever we have pyruvic acid, which is the end product of the glycolysis, so that pyruvic acid under the anaerobic conditions undergo it change either into lactic acid or alcohol or carbon dioxide. So carbon dioxide is a byproduct during the process of fermentation. So in that process, when carbon dioxide is released, it will make the dough puffy. Okay, I hope you got the point. Now after that, similarly, the dough which is used for making bread is fermented using baker's yeast. Now baker's yeast is another name for the yeast, which is Saccharomyces cerevisiae. This is the name of the yeast. Okay, and this is the name of the yeast, and actually it is useful in making bread. Okay, because bread has also to be puffy and spongy. So who does that? The yeast, which is Saccharomyces cerevisiae. A number of traditional drinks and food are also made by fermentation. Yes, you would have seen beer, wine, whiskey, rum, anything. So they all are nothing but whatever juices you can see, those all are produced by the fermentation process only. A toddy, which is a traditional drink of some parts of southern India, is made by fermenting sap from plants. So the sap is collected from the plants, and that sap is undergone fermentation and produces toddy in southern India parts. Each restriction into, okay, so after that we have, now there are microbes which are also used to ferment fish, soybean and bamboo shoots to make foods. Cheese is one of the oldest food items in which microbes were used. Now there are also microbes available in our household. With the help of those microbes, what we can do is we can ferment our fishes, soybean, bamboo shoots. And why we are doing it so? Because to make food. Cheese is one of the oldest food items in which microbes were used. Okay, so whatever cheese we are preparing at our homes, that is because of the because of the action of the microbes itself. Different varieties of cheese are known by their characteristic texture, flavor and taste. The specificity coming from the microbes used. Now whatever microbe you will be using, because you can use different microbes in order to make cheese, so whatever microbe you will use, that depend at how your cheese will form, what kind of cheese you will be able to prepare. Okay, now next is, for example, the large holes in Swiss cheese. Now if you have seen Swiss cheese, just check it out on Google. So in Swiss cheese we have lot of holes, like if you have seen Tom and Jerry cartoon, in your childhood times, so you have seen that they always show Swiss cheese. Now that's Swiss cheese prepared due to the production of large amount of CO2 by a bacterium named as Propionibacterium sharmani. Okay, so this bacteria is responsible for the production of Swiss cheese. The Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. So fungi is again a microbe, okay, a microorganism, or fungi is basically a part of your kingdom plantae. Okay, uh, fungi is a part of your kingdoms. Okay, kingdom fungi. So I'm sorry, it's not a part of kingdom plantae, so I excuse you for that. It's a different kingdom. So Roquefort cheese is prepared when a fungi is grown on the cheese itself, so that it will give a particular flavor. Next we have microbes in industrial products. So beverages and antibiotics are some examples. Production on an industrial scale requires growing microbes in very large vessels called fermenters. Now in industries, obviously industries are very large scale setups. So whatever we are preparing, whatever products we have to get, we have to use large amount of microbes in that. And whenever we are doing any fermentation in industries, fermentation is always done into fermenters. Okay, so fermenters are the big tanks situated in your industries. Fermented beverages. Now how are we able to prepare beverages? Microbes, especially yeast, that is Saccharomyces cerevisiae, has been used from time immemorial for the production of beverages like wine, beer, whiskey, brandy or rum. For this purpose, the same yeast Saccharomyces cerevisiae used for bread making are commonly called brewer's yeast or baker's yeast, because it can also used in baking, it can also be used in fermentation or preparing beverages. So it is also called brewer's yeast. It is used for fermenting malted cereals and fruit juices to produce ethanol. Okay, so as you can see that in whatever beverage we have, whatever for example, if we have wine, beer, whiskey, we have alcohol in there. So alcohol is nothing but the ethanol which we have. Along with that ethanol, we have carbon dioxide with us. Reaction which is taking place in fermentation by yeast. So what is the reaction? If I just explain you again, in glycolysis cycle. So if I just show you the glycolysis, in glycolysis cycle, what was happening? Glucose was getting converted to pyruvic acid under the action of many enzymes, and many energy bioenergetics was taking place over there. Now this pyruvic acid, okay, this pyruvic acid, if anaerobic condition, okay, if in anaerobic condition, and the enzyme which is getting used up over here is dehydrogenase. So it is basically producing the plus decarboxylase. Okay, so what is it producing? It is producing ethanol plus CO2 release. CO2 is getting released. Okay, now also that there is what, if I tell you the bioenergetics, so NADH plus H positive molecule will convert into NAD positive molecule. So this is the whole process of pyruvic acid, which is producing ethanol, which is the fermentation process. So similar thing I have written over here, and this is done how? With the action of yeast. Okay, which yeast? We have Saccharomyces cerevisiae. Okay, this is the yeast. Next we have, depending on the type of the raw material used for fermentation and type of processing, with or without distillation, different types of alcoholic drinks are obtained. Yes, distillation is nothing but distillation is a type of or series of processes through which the remnant or the other material from the drink is removed, and only the alcohol and the other thing is obtained, like the suitable thing is obtained, whereas other things are removed. So this is what I was able to explain. These are the bigger tanks called fermenters available in industries, and this is the whole setup. This is a whole setup of everything in which fermentation goes on and we prepare beverages and everything. So this is fermentation plants. So for juices, whatever you see, Natural Bee, you can see, different juices brand names. They all have these type of setups or the fermentation plants. Okay. Wine and beer are produced without distillation. Now, distillation means filtration in some way. So, wine and beer are produced without filtration, and whiskey, brandy, and rum are produced with distillation of the fermented broth. So, whatever fermentation is done, whatever fermentation is happening, the end product of fermentation, okay, end product of fermentation will consist of lot of materials, like the main material which which wanted to prepare, that will up there, the enzymes, the microbes will be there. So end product will be called as fermented broth. Okay, so wine and beer have this broth, whereas whiskey, rum and brandy, they do not have this broth. Okay. Antibiotics. So antibiotics, now let us we talked about beverages, now we talk about antibiotics. So antibiotics are, anti means against and bio means life, but whose life they are against? That they are against the life of disease causing microbes. Okay, this is a very important point you need to remember, so that you will not get yourself laughed at. Disease causing organisms. So antibiotics are basically those medicines, you can say, which basically help to kill that disease causing organism and to help to save the life of the host. Okay, together they mean against life, but when it comes to the context of disease causing organism, whereas if we talk about human beings, so they are pro life, it means they support the life. Okay, it means they are supporting the life of the human being. Okay, next is, antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of the other disease causing microbes. Yes, so they are chemical substances. Commonly used antibiotics is penicillin. This is very important. Penicillin is the first antibiotic which was discovered or prepared. Okay, so this is again an important point. You may be asked in the NEET exam, which was the first antibiotic which was available? So you can simply say penicillin. Okay, next we have Alexander Fleming, while working on Staphylococci bacteria, once observed a mold growing in one of his unwashed culture. For example, there was a scientist called Alexander Fleming who actually discovered, so I will write over here, he discovered what? Penicillin antibiotic. Okay, so this question is again important. They will ask you for the name. So the name is Alexander Fleming. Now, how did he got penicillin? Basically, he had so many culture plates, and on those culture plates, the Staphylococcus bacteria was not growing. Why it was not growing? Because penicillin was there. Okay, so then the final name, which was and the name of the antibiotic was penicillin, whereas the mold, that is the fungi, was Penicillin notatum. So this is a member of fungi kingdom. Okay, Penicillin notatum. However, its full potential as an effective antibiotic was established one later by Ernest Chain and Howard Florey. But Alexander Fleming was not able to, like explain the benefits of that antibiotic, wherever the benefits were explained by these two scientists, Ernest Chain and Howard Florey. Okay. Next we have, antibiotics have greatly improved their capacity to treat deadly diseases such as plague, whooping cough, diphtheria, and leprosy. So these are some deadly diseases. These are not deadly diseases nowadays, but before, when the antibiotics were not discovered or prepared, then they were the deadly diseases. Why? Because Plague was causing by rats, in which there was an infection, and it will ultimately lead to the death of the individual. Whooping cough is something, let's discuss them out. In whooping cough, there is a forceful inspiration after coughing. For example, you are coughing and you are getting breathless, so you have to inhale so much of air. It means you are making some pitchy noise, okay, like like this, okay. So that will be called as whooping cough, okay. So it was deadly because it was getting happened or occurred in children. So children were not able to inhale that much of air, so it led to the death of them. In diphtheria, diphtheria is another type of disease in which there is a formation of white leathery membrane in the in the pharynx. For example, if this is the mouth and this is the tongue, these are the teeth, this is the tongue over there. So in the soft palate, lying at the pharynx, there was a formation of this leathery membrane. This leathery membrane was actually covering your pharynx, and you can imagine that pharynx is a common passage of food and air. If pharynx will be covered by this unusual membrane, then you will not be able to engulf food, or either air will not get into the larynx. So that's why it was very severe condition, and it was also leading to other further severe complications. Leprosy, on the contrast, is a contagious, it is a highly contagious disease. It means contactable or communicable disease that affects your skin, mucous membrane, nerves, causing discoloration and lumps on the skin. In severe cases, disfigurement and deformities are also there, okay. So leprosy was also a deadly disease at that time. So leprosy is now mainly confined to tropical Africa and Asia only. Chemical enzymes and other bioactive molecules. Now, what are the benefits we are getting from microbes being a chemical or an enzyme and other bioactive molecule? Microbes are also used for commercial and industrial production of certain chemicals like organic acids, alcohols, and enzymes, okay. Example of acid producers are Aspergillus niger, which produces Aspergillus niger, which produces citric acid. It's a fungus, so it's a microorganism which is producing your acid, okay, which is actually beneficial for your daily living or your industrial purposes, or any chemical purposes. So Aspergillus niger is producing citric acid, Acetobacter aceti is producing acetic acid, Clostridium buticulum is producing butyric acid, and Lactobacillus will produce lactic acid. So you can remember this because these can again come into your NEET exam. This is important. Then microbes are also used for the production of enzymes like lipases. Lipases are the enzymes which are basically help in degradation of fats. Okay, so you can also like lipases are also formed by none other than enzymes also. Lipases are the enzymes which are formed by microorganisms. Now, lipases are, where are they used? They are basically used in their detergent formulations and help in removing oily stains from laundry. Okay, so because these are fats and these are hydrophobic, that's why they are applied to the cloth, so that they stick to the cloth and to the stain whenever we apply water. So they will get removed due to the action of the water being a hydrophobic in nature. So they get removed along with the oily stain with them. So this is the action of your stain removal. Bottled fruit juices bought from the market are clearer as compared to those made at home. This is because the bottled juices are clarified by the use of pectinases and proteases. Okay, whatever juice you make at home, that juice doesn't seem to be very much cleared, but the bottle juice you get, that really is very cleared and pure because of the presence of the microorganism called and because it is clarified by the use of pectinases and proteases. These are again enzymes which are responsible for the degradation of proteins, okay, proteins, peptones, and proteoses. So these are the these are the proteins, you can say, and they act upon this and convert them into amino acids. Okay, basically proteins are getting degraded. So in order to filter the bottle juice, these enzymes are also used, which are a product of your you can say the microorganism itself. Now it's important again, this you have to remember, streptokinase, which is produced by a bacterium streptococcus and modified by genetic engineering, is used as a clot buster. So streptokinase is again an enzyme which is produced by the bacteria called streptococcus. It's a bacteria and it is modified by genetic engineering, used as a clot buster, means it helps to remove the clots from blood vessels of patients who have undergone myocardial infarction leading to heart attack. For example, there is an artery and there is any block in over here. So what will we give is streptokinase. Is present in medicines. This is given to the patients so that the clot can be removed from there. Okay, and this is, and if the clot will not get removed, then we will get heart attack. In scientific language or in medical language, we call it as myocardial infarction. Okay, so I hope this is clear to you. So streptokinase is important. Another bioactive molecule called cyclos is again important. Cyclosporin A, that is used as an immunosuppressive agent. So we have cyclosporin A, which is again we have a product obtained from the microorganism. It is responsible for the immunosuppression whenever we are doing any organ transplant, and it is produced by the fungus called Trichoderma polysporum. So this is a microorganism, and this organism is producing cyclosporin A, which is helping in immunosuppression, okay, in organ transplants. So as you can see, you have antigen antibody reactions. Whenever you are doing any organ transplant, you would have studied in genetics that we have antigen antibody reaction. You cannot just, being a doctor, can go, like for example, if somebody needs any organ, for example kidney, so you will just get any kidney out of the patient and just transplant. No, you have to match the antigens and antibodies. So how are they matched? And in order to transplant the organ, how can we suppress the immune response of that? Because these are nothing but the result of your immune response, right? Because your T lymphocytes are responsible. You can say T lymphocytes are present for the immune response, and the response is called as humoral response. Okay, so in order to suppress this immune response, we give this cyclosporin A. Okay, next we have statins. This is again important. Statins produced by the yeast Monoscus purpurus have been commercialized as blood cholesterol lowering agents. So whenever a patient has a high amount of cholesterol, we give that patient statins, which is produced by the yeast Monoscus purpurus. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So whatever enzyme we have for the synthesis of cholesterol, the statins will prevent that enzyme to produce cholesterol, so that the person will have a healthy liver. Okay, because you can see the cholesterol, we have many types of cholesterol, so those cholesterols are basically produced and stored in liver itself. Microbes in sewage treatment. Now we talked about microbes in industrial products, microbes in household products. Third, we are going to discuss is microbes in sewage treatment. So in sewage treatment, how can we do so? The large quantities of wastewater are generated, and every day in cities and towns, a major component of this wastewater is human excreta. You would agree with me that whatever wastage we have today, we have a lot of wastage. You can see the population of the whole world, but not India, but the whole world. So you can see how much excreta, whether it be it you can see a urine, or be it a poo or whatever, a poop. So whatever we have, the excreta is the major poisonous, you can say major polluting component we have these days. This municipal wastewater is called as sewage. Now whatever excreta is flowing into the sewages, that is called as sewage. It consists large amounts of organic matter and microbes, many of which are pathogenic. Yes, so obviously, like we are organic in nature, our body consists of so many organic compounds. So similarly, the excreta will also be organic. It will consist of organic materials and microbes. And those microbes can be pathogenic. They can be disease causing to anyone. For example, to the animals, to the plants, to the other things. Okay. So how can we prevent, like now what happens, this cannot be discharged into natural water bodies. Absolutely, the excreta what is being produced, or any other you can say the debris is produced, we cannot just flow that debris or that excreta into the river body, the water bodies like rivers, oceans, seas, streams, whatever. Before disposal, we have to clear all the debris, all the pollutants, all the pathogens out of that, so that there will be no harm to the environment. So how can we do so is, we have sewage treatment plants. These are very important, STPs, sewage treatment plants. Now what has happened into these plants, we will discuss it. Treatment of wastewater is done by heterotrophic microbes naturally present into the sewage. Okay, so in a sewage there are microbes which are heterotrophic in nature. It means they get their nutrition from that excreta or that debris. So some of them are degraded into the sewage itself by these microbes, so they are useful for us. This treatment is carried out in two stages. Now the treatment which I am talking about, the heterotrophic microbes degradation, this is happening into two ways. The first will be the primary treatment. Primary treatment is something, these treatment steps basically involve physical removal of particles. Like somebody, or we, with the help of machines and the technology, we can physically remove those particles out of that sewage, the wastewater, large and small, from the sewage through filtration and sedimentation. How can we do? We can either filter, we can also filter that out, and we can sediment the higher particles, so that the clear the clear slur, or the clear filter will be able to get on the surface. So filtration and sedimentation are the processes of the primary stage. These are removed in stages. Initially, floating debris is removed by sequential filtration. Whatever debris we have floating, which is floating on the surface, we actually filter it out. We have many types of filters. For example, for big particles, we have big filters. For small particles, we have filters with very minute, filtration, you can say sheets. Then the grit, soil and small pebbles are removed by sedimentation. Yes, whatever whatever soil and pebbles we have, stones we have, they all are sedimented. They all remains on the bottom, wherever the other filtrate will comes on the surface. Then all solids that settle from the primary sludge and the supernatant forms that the effluent from, okay, whatever primary sludge we get after the filtration and sedimentation process, that is called as effluent. Okay, you just remember this, effluent. The effluent from the primary settling tank is taken for the secondary treatment. Now this effluent is actually getting ahead for the secondary treatment. Okay, now what happens? Secondary treatment or biological treatment, in this what happens, the primary effluent is passed into larger aeration tanks where it is constantly agitated mechanically and air is pumped out into it. What happens is, now there are very big tanks. Okay, those big tanks consist of larger amounts of air, oxygen. Okay, so what happens basically, and there is a constant agitation. What is agitation? That is mixing up of that primary sludge continuously mixing and high mixing, like mixing with high intensity. Why we have to mix it? So that it will mix out with the air which is getting pumped into the tanks. Okay. So now what is the benefit? Why are we mixing the oxygen with that primary sludge? This allows vigorous growth of useful aerobic microbes into flocks, that is masses of bacteria associated with fungal filaments, to produce mesh-like structures. So, like, because see, we are providing oxygen to the tanks, it means we are creating an anaerobic environment. Sorry, aerobic environment. Aerobic means presence of oxygen. So we are preparing an aerobic environment. Whenever an aerobic environment is there, it means that it will lead to the the growth of the aerobic microbes, and that microbes, they are helpful to us because they will destroy the harmful microbes from that primary sludge. Okay, how will they do it? Because whatever bacteria will be present into the primary sludge, they will make association with the fungi, and that association will be called as flocks. So flock formation will be there. Okay, while growing, these microbes consume the major part of the organic matter in the effluent. Now, whatever organic matter we have, okay, we have so many pollutants which are organic in nature. So these microbes will engulf all that organic material, the majority. This significantly reduces the biological oxygen demand, biochemical or biological oxygen demand. Biochemical oxygen demand means the oxygen demand which is required by those microbes present in 1 liter of sludge or slurry. Biological refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. For example, there is a container. We have 1 liter of the water and the organic matter mixed with it. Now, whatever bacteria will be present in there, whatever oxygen they will consume, that oxygen will be called as biochemical oxygen demand. Okay. So in order, we have to decrease this oxygen demand because oxygen we need for other processes. Why do we have to waste this oxygen on these microbes? So basically we have to decrease the amount of the organic matter. How are we able to do it? We are introducing aerobic bacteria into there. So they are engulfing that organic matter. As a result, if organic matter is decreasing, it means ultimately the biochemical oxygen demand will also get reduced. Okay. The BOD test, the biochemical oxygen demand test, measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus indirectly BOD is a measure of organic matter present into the water. So because see, microorganisms are also living, they also need oxygen, so they will get the oxygen, but we have to decrease their amount so that they will not get the oxygen, because oxygen is required for other processes. So how are we able to do it? We are decreasing oxygen of organic matter. The greater the BOD of wastewater, more is its polluting potential. Yes. For example, if this is a container and this is a container, okay, if BOD is more in this, it has some BOD, it has some BOD. Okay. Now, how will you decide which container contains more polluting material? This one, because the BOD is directly proportional to polluting potential. Okay. So if BOD is more in container A, then the polluting material or the potential will be more in this. So PP, I am writing PP, that is polluting potential, will also be in greater in A container. So this is how you can decide it. Once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into a settling tank. Now, in the secondary treatment, when the primary sludge was acted upon by the microorganisms, so once the BOD, the biochemical oxygen demand, gets reduced, then what happens? The effluent or the remnant is passed into a settling tank where the bacteria flocks are allowed to sediment. Okay. Now again, they are they are placed into those tanks where these flocks are sedimented, so that only we have is left out that, or that filtrate. This sediment is called activated sludge. Now we had primary sludge, now we have activated sludge. A small part of the activated sludge is pumped back into the aeration tank as an inoculum. Inoculum is, again I am telling you, it is a starter which initiates the process, so that the which actually contains a lot of microbes, and that microbes will lead to the production of new microbes. Okay, so inoculum is a starter. We are also using we are taking a small part of the secondary sludge, and we are putting that sludge into the, you can say, into the aeration tanks where the primary sludge is there. The remaining major part of the sludge is pumped into larger tanks called anaerobic sludge digesters. Okay, what happens is, here other kinds of bacteria which grow anaerobically. Now aerobic part is done. In aerobic part, microorganisms which were aerobic, they ate up, they engulfed up all organic matter. But what happens with anaerobic matter? That matter which consists no oxygen, so they have to be removed by the anaerobic bacteria. So basically we are we are settling that secondary sludge into the other tanks where we provide anaerobic conditions. So the anaerobic bacteria digest the bacteria and the fungi in the sludge. During this digestion, bacteria produce a mixture of gases called methane, hydrogen sulfide, carbon dioxide, that is CH4, H2S, and CO2. They are present over there, and what they will do is, they are the gases, they are called biogases, that is, they actually can help as a fuel. So these are called biogas, and they have been used as a source of energy. It is inflammable because they can inflamed, they can trap the fire. Okay, they can catch fire. That's why we call them as inflammable, and they are biogas. They provide no harm to the environment other than your global warming, and they can be used in many household uh, processes. The effluent from the secondary treatment plant is generally released into the natural water bodies like rivers and streams. Now whatever remnant we have after doing all these processes, whatever filtrate we have been obtained, we flow that filtrate into the water bodies, that is rivers, oceans, seas, lakes, whatever. Okay, so now so that the water bodies will not get polluted, and the life in the water bodies, that is fishes, we have different sea different water organisms, they all will remain safe. Okay, they will not die. That's why we have to do this all process. And when we talk about humans, so again, okay, so we are going to get the filtered water at our homes. Now if there will be any microorganism available in that, that will serve as a pathogen and a disease causing organism. Now due to increasing urbanization, it means more population, sewage is being produced in much larger quantities than ever before. However, the number of sewage treatment plants have not increased. Sewage is increased, but sewage plants are not increased. So what happens is, untreated sewage is often discharged directly into rivers, leading to their pollution and increase in waterborne disease. Absolutely, you can understand, if that sludge or slurry will not get filtered out, if all the excreta will not get filtered out, so there will lead to a lot of waterborne diseases. As you can also imagine, like we have diarrhea. Okay, we have other diseases which are related to water, like typhoid, like, right? Or we have a lot of diseases like amoebic dysentery. Okay, so the Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save people, to save these major rivers of our country from pollution. Now these are some ministries which have taken the responsibility in order to protect the environment and the water bodies. Under these plans, what happens is, it is proposed to build a large number of sewage treatment plants so as only treated sewage may be discharged into the rivers. Yes, so what they have initiated is, they will build more treatment plants so that the water bodies will not get polluted and waterborne disease will not take place. Now we will talk about microbes in, okay, biogas. Now we'll talk about biogas. So what is biogas? Biogas is a mixture of gases containing predominantly methane, produced by a microbial activity and which may used as fuel. Okay, as I told you, it's highly inflammable, so it can be used as a fuel as well. This type of the gas produced depends upon the microbes and the organic substrates they utilize. Whatever microbes will be used, and whatever substrates those microbes will be used, they will decide what kind of biogas we are going to have. Either we are going to have CH4, we are going to have carbon dioxide, or we are going to have hydrogen sulfide and many other biogases. Okay, in the examples it cited in relation to fermentation of dough, cheese making and production of beverages, the main gas produced by CO2, you can understand because I explained you the process of fermentation. So CO2 was a byproduct. So in all fermentation processes, CO2 is getting produced. However, certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with CO2 and H2. Okay. So whenever there is a bacteria which grow anaerobically on cellulosic material, these bacteria are collectively called as methanogens. Okay, why they are called as methanogens? Because they are producing methane. And one such common bacteria which is called as methanogen is methanobacterium. You just need to remember the name. It can come into your question in NEET: methanobacterium and methanogen. Whenever there is any word gen, it means there is a production of methane, and methanobacterium, as the name suggests, is a bacteria which is working as a methanogen. These bacteria are commonly found in the anaerobic sludge during sewage treatment. These bacteria, being an anaerobic bacteria, they are actually required in the last step of the sewage treatment plan. These bacteria are also present in rumen, a part of stomach of cattle. So if you have heard of, if you have ever seen buffalo, so in the stomach of buffalo, there are, lot of chambers in the stomach, 4 chambers in the stomach, and the cattle consist of rumen. Okay, that rumen is a part of the stomach, and in that part what we have is these methanogens. What they do is, a lot of cellulosic material present in the food of cattle is also present in the rumen. So these kind of bacteria, they help in the breakdown of their cellulose and help in digestion of the cellulose, giving nutrition to the cattle. Okay, so they have these methanogens with them. That's why in a cow dung we get these methanogens out. Okay, because they are present in their stomach. Thus the excreta, dung of cattle, commonly called gobar, is rich in these bacteria. Dung can be used for generation of biogas, commonly called as gobar gas. Okay, next we have is, can humans digest cellulose? Now there's a question which arises, can we digest cellulose? Because we are eating a lot of plants, right? Whatever, for example, whatever vegetables we are eating, they are all plants, so we get cellulose in some manner. So do we digest that cellulose? The answer is no. We cannot digest cellulose because we do not have any bacteria available to digest it. Okay, so that's why, guys, you are preferred whenever someone is suffering from constipation or upset stomach or whatever, someone is not able to excrete properly, because their intest so why is it so? Why they are advised to have fibers? Because cellulose is nothing but a fiber, and fiber is available in fruits, vegetables. That's why the doctors always say that eat fibrous fruits, because fibers are non-digestive in nature by the humans, and that's why they make the removal of the excreta easy, into your intestine. You can see they keep food moving through the gut and push waste out of the body easily. Okay, that's why it prevents constipation and all. So I hope it's clear that cellulose cannot be digested by, the main point is cellulose cannot be digested by humans. Okay, second point says fibers should be taken in diet. Always just remember this: fibers are non-digestive in nature by humans, so that's why they are prescribed by the medical professionals to be taken into your diet. The biogas plants consist of a concrete tank. Now, how does a biogas plant look like? It basically consists of a concrete, that is a brick tank, which is 10 to 15 feet deep, in which bio wastes are collected. Whatever we have, for example, cow dung or any other organic material consisting of, carbon dioxide and other things. It is basically are collected into those tanks, and a slurry of dung is fed. Okay, it's a form of a slurry. So a highly viscous fluid, a floating cover is placed over the slurry. Why? Because which keeps on rising as the gas is produced. Whenever the gas is produced, the cap will rise up due to the pressure of the gas. So it will indicate that yes, that cow dung is actually producing your biogas. So how it is producing? Due to the activity of microbial activity. Which microbium, which microbe, as I told you, okay, the microbe in the previous slide. The biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. So as you can also see that at your home you are able to get LPGs. So these are nothing but the biogas which you are able to get directly from the biogas plants through pipes. Also, the spent slurry, whatever slurries remained over there after the biogas formation, that is used as manures and fertilizers into fields. Okay. Cattle dung is available in larger quantities in rural areas where cattle are used for a variety of purposes. Okay. So biogas plants are more often built in rural areas. The biogas thus produced is used for cooking and lighting. Okay. So the gas which is produced, it's highly flammable in nature. That's why it catches fire, and that's why we are able to cook food, and also for the lightning purposes like burning lamps and everything. Now this is how a biogas plant looks like. So you can see that over here we are giving the slurry or the sludge, the cow dung. This is getting stored into this digester, which is 10 to 15 feet long. A gas is formed after several days. These are the gases: methane, carbon dioxide, or whatever gas we are. This is a pipe which I was talking about. It is getting to your homes, and it is producing it is making the biogas available to you. The left remnant is gas holder sludge, which is actually will work as manure and fertilizer into the crop fields. Okay. So next what we have is the technology of biogas production was developed in India itself, mainly due to the efforts of Indian Agricultural Research Institute IARI and Khadi and Village Industries. So these are the two which actually discovered or actually responsible for the formation of these biogases. Next we have microbes as biocontrol agents. Now let us talk about those microbes which are useful as biocontrol agents. What is the biocontrol agent? Biocontrol refers to the use of biological methods for controlling plant diseases and pests. Okay. So whatever diseases are caused in plants due to the pests and insects, whatever, those are, the controlling or preventing those pest to infect the plants is called as biocontrol. Okay. Now the agents which are responsible for biocontrolling is biocontrolling agents. These are chemicals are toxic and extremely harmful. Yes, biocontrol agents are harmful to human, sorry, not biocontrol, but the pesticides, insecticides, whatever we are using, they are very harmful to the nature, to human beings, to animals alike, and have been polluting an environment, soil, groundwater, fruits, vegetables, crop, plants. Yes, whenever you are giving any pesticide or insecticide, what you are doing, that will actually come into the plant through the roots, you write xylem phloem function, and in plant parts like flower, fruit, stem, leaves, branches, in every part of the plant, and we are eating those things, right? So ultimately it's creating a chain. If a herbivore is eating, then it will go into the body of the cattle or any animal. If we are eating, it is going into our body. So if that is polluting the plant, ultimately it will pollute the body of the other organisms, whoever they are, whoever are eating that plant. Okay. Next, we have biological control of pests and diseases. So in agriculture, there is a method of controlling pests that relies on natural predation rather than introducing chemicals. Now in agriculture these days, the farmers are really educated by our institutes, by our government, on how to use these biocontrol agents. Because in earlier times, what used to happen is, whenever in a field a pest or any microbe or any other organism is producing, they would really think that it's going to harm their crops, and they would just kill that by the use of pesticides and insecticides. But nowadays they are made aware by the government that all the pests, all the microbes, all the organisms are not harmful to the nature. Many of them are also useful to the crop fields, right? They help in the growth of the plants, they help in the faster growth of the plants. So they do not have to use pesticides, insecticides, weedicides, fungicides all of the time. Okay. So you can see a key belief of the organic farmer is that a biodiversity further health. A more variety of landscape has, the more sustainable it is. Okay. The organic farmer therefore works to create a system where the insects that are sometimes called pests are not eradicated. That's what I meant. There are organisms like insects, microbes, or whatever, they are getting produced into the fields, but still the farmer do not eradicate them, because the farmer is educated enough that they will be able to correlate the relationship between the insect and the plant, because insect will help the plant, and in return plant will also help the insect. We'll study how. So what they are educated about is to manage the levels by a complex system of checks and balances within a system and vibrant ecosystem. Contrary to the conventional farming, now in conventional means older farming methods I explained you, what they used to do is they use chemical methods to kill both useful and harmful life forms indiscriminately. This is a holistic approach that seeks to develop an understanding of the webs of interaction between myriad of organisms that constitute field flora and fauna. The use of biocontrol measures will greatly reduce our dependence on the toxic chemicals and pesticides. An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as pests, and also their life cycles, pattern of feeding, and the habitat that they prefer. They will help develop appropriate means of biocontrol. So biocontrol is nothing but understanding the advantage of keeping or managing the levels of those pests, those you can say predators, those their life cycles and everything, so that they will in future give some benefit to the growth of the plant or the field. This is very familiar beetle. Okay, now you would have seen that a beetle with red and black spots, the ladybird, we call that beetle as the ladybird, and the dragonflies are useful to get rid of aphids and mosquitoes. Now the farmers are educated to keep keep these beetles and dragonflies into their crop fields, because they actually help to kill aphids and mosquitoes respectively. An example of microbial biocontrol agent that can be introduced in order to control butterfly caterpillars is the bacteria Bacillus thuringiensis. Now, there is a microbe which is useful in order to control or prevent butterfly caterpillars from engulfing the plant parts is by Bacillus thuringiensis, that we call as BT. B stands for Bacillus and T stands for thuringiensis. This is the name of the microbe or the bacteria. Now this BT is available in sachets as dried spores. Now you can get it from the market. Now how are they useful is, they are, first of all, you can get it in the form of sachets as dried spores which are mixed with water and sprayed onto vulnerable plants. So what you have to do is nothing but to buy a packet of these BT, and then you can just mix them with the water and put them into the plant, spray them onto the plants such as brassicas, and you can see fruit trees, which these are eaten by the insects larvae. So the larvae of these butterfly caterpillars, they actually eat the plant parts, which no farmer would want. So the farmer is educated to buy these packets and prey onto their fields. In the guts of the larvae, the toxins is released and the larvae gets killed. Okay, so whenever a larvae engulfs these Bacillus thuringiensis bacteria, this is highly toxic to them and they ultimately kill that larvae. The bacterial disease will kill the caterpillars but leave other insects unharmed. Okay, so they are only restricted to bacterial caterpillars, they will not harm the other useful organisms. The development of methods of genetic engineering in the last decades or so has made the scientists have introduced B thuringiensis toxin genes into plants so as to prevent the plants from harmful organisms or insects. Some plants are resistant to attack by insects pest. Bt cotton is one such example. Now there are some plants which have already an resistance to attack by insect pest because of the Bt cotton. Like a Bt cotton is one such example in which cultivated because in Bt cotton you can see in a cotton plant we have already introduced Bt through genetic engineering. Okay, we have introduced some genes of Bacillus thuringiensis into the cotton plant so that the plant has developed the resistance against these pests or the insects. Okay, a biological control being developed for use in the treatment of plant disease is the fungus Trichoderma. Trichoderma species are free living fungi that are very common in root ecosystem. They are effective biocontrol agents for several plant pathogens. So there is a fungi called Trichoderma polysporum. What that does is they basically help or remain in the association of the roots in the plants, and they help the plant against several plant pathogens. Okay, like the harming of the plant by the pathogen. Now, baculoviruses again are pathogens that attack insects and other arthropods. There are baculoviruses, which are again biocontrol agents. They attack insects and other arthropods, which actually, like kill the plants or you can say eat the plants. The majority of baculovirus used as biocontrol agents are genus nucleopolyhedrovirus. This is a main biocontrol agent or baculovirus, we can say. Biocontrol agent. So this is a baculovirus. Okay. Next we have these viruses are excellent candidates for species specific narrow spectrum insecticidal applications. So basically these viruses will not infect the other organism. They are very much restricted to a class of organisms, okay, a class of species. They have been shown to have no negative impacts on other plants, mammals, birds, fish, or even or no targeted insects. Microbes are biofertilizers. There are problems associated with the overuse of chemical fertilizers, and there is a large pressure to switch to organic farming. So see, by even using of the usage of the fertilizers in fields is not very much essential, because fertilizers in some way or the other they also affect our health. So we use biofertilizers. What is biofertilizer? Basically the fertilizer which are not chemical in nature but they are biological. It means, for example, if you would understand, whatever cow dung, whatever sludge or whatever remnant is we have after the formation of biogas, that is totally organic in nature. That is not going to produce any harm to your plants, to your fields. So those are called biofertilizers. Okay, and the use whenever in a plant we use only natural methods. We do not use any chemicals, we do not use anything. That is called an organic farming. Okay, means pure farming. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main source of biofertilizer are bacteria, fungi, and cyanobacteria. If there will be a pure farming, organic farming, if there will be use of biofertilizer, then yes, the quality of the plant and the quality of the soil will also be improved. That's why the farmers are educated and advised to use biofertilizers into their crop fields, and they do so. The nodules on the roots of the leguminous plants. Now, leguminous plants are nitrogen fixing plants. In the roots of those plants, there is symbiotic association of Rhizobium bacteria. Rhizobium is a bacteria and it remains in the association with the roots of these leguminous plants. These bacteria fix atmospheric nitrogen into organic forms. Okay. So whatever the nitrogen is present into the atmosphere, it is gets fixed into the organic forms, which is used by the plant as a nutrient. Okay. That is called nitrogen fixation. Other bacteria can fix atmospheric nitrogen while free living in the soil. Example, Azospirillum and Azotobacter. So rhizobium has to be in the association with the roots. Rhizobium association, that is bounded. Okay, it cannot freely move. Whereas Azotobacter and Azospirillum, okay, they are free living. They can freely move into the soil and still they are able to fix the nitrogen, okay, for the plant. Fungi are also known to form symbiotic associations with plants mycorrhiza. Now there is again a symbiotic association of a fungi with a plant, which is called mycorrhiza, in which what happens, many members of this genus Glomus form mycorrhiza. The fungal symbiont in these association absorb phosphorus from soil and pass it to the plant. Because phosphorus is again a nutrient for the plant, so phosphorus has to be reached to the plant from the soil. And how will it be done is, the fungi, they are in the association with the plant, they form the association with the plant, and then they help to absorb the phosphorus into them and pass that phosphorus to the plant so that the plants get nutrition. Okay, so it's again an important point for you all. Plants having such association shows other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought, and overall increase in plant growth and development. So whatever bacteria we have studied, whatever fungi we have studied who remain in association with the plants, they give nutrients to the plants. So the plant, first of all, the plant will become resistant to root-borne pathogens. Plant will also tolerate it to the salinity. It means high amount of salt present into the soil, and the drought. Okay, so they will be healthy and nutritious plants. The fungi obtained carbohydrates produced by the plants through roots, and the plants receive nutrients and water from the fungal association. Now, what the benefit the fungi is having in order to be into the association with the root of the plant? Fungi is having nothing but the fungi is able to get carbohydrates from the plants. So fungi is also growing, plant is also growing. The hyphae absorbs nutrients and water from the soil and make them accessible for plant. This way both species receive the benefit. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen, Anabaena, nostoc, oscillatoria, etc. Now, cyanobacteria is also a part of your bacteria only. It's a blue-green algae, also called as BGA. Okay? Blue-green algae. So, the cyanobacteria, they are the autotrophic. It means they prepare their food on their own. They are the microbes. They are widely distributed in aquatic and terrestrial environments. They live in both, that is water and earth or land, many of which they can fix atmospheric nitrogen again. That means they will trap the atmospheric nitrogen and convert them into the nitrogen beneficial for the growth of the plant. And what are these cyanobacteria? Anabaena, nostoc, and oscillatoria. Okay, so you need to remember that point as well. The next is in paddy fields, that in the fields where the rice is grown, cyanobacteria serves as an important biofertilizer. In blue-green algae also add organic matter to the soil and increase its fertility. Again, in paddy fields, these cyanobacteria are used so that they will help in nitrogen fixation and ultimately increase the fertility of the soil also. Okay, so the soil also gets improved in the quality, and the soil becomes more fertile. That is, more plants can grow onto that soil easily. Okay, so guys, this was all about today. I discussed the whole chapter, microbes in human welfare. We had studied about what kind of microbes are available into, so that they are helpful for the welfare of the human beings. Because you can see whatever we had studied, be it a household, industrial, sewage treatment, or in the formation of antibiotics, everything that was only and only beneficial for us, right? So microbes are, you should not always think that whatever microbes are surrounding us, they are all harmful. No, we have defense mechanisms available in our body which are protecting us from those microbes. But in addition to that, there are a large number of variety of microbes which are available, which are also very much useful for our bodies, for our better functioning, right? For our other purposes. So so I hope you were able to get something out of this chapter, and this chapter will be easy for you when you will study this chapter on your own. So you, in order to access this chapter in this lesson, you can just go through the YouTube videos at any time. Thank you so much guys, and I wish you best of luck again, and be safe in this pandemic, and take care. Thank you so much. Bye bye.